Hi, I'm Lindsay Farrer, and as chair of the session on tau and neuronal signaling, I'm going to introduce you to some of the genes that are involved in this pathway, and in particular, focus on a couple of them that I can show you how we go from gene association to understanding disease mechanism related to those genes. Here is a Manhattan plot showing the results of a large recent genome-wide association study. And you can see that there are a large number of genes, over 25. And in this particular study led by Brian Kunkel, 5 additional genes were identified. But I want you to focus not on what you're seeing, but on what you're not seeing. What's missing from this plot? Well, for starters, we're missing MAPT, the gene that encodes tau. Why is it that we have unable to identify association with a gene that encodes one of the critical proteins involved in Alzheimer's disease? A couple of years ago, my colleague Gyunga Jun led a study for the International Genomics of Alzheimer's project, specifically at looking at association within subgroups of subjects, those with and those without ApoE4. And one of the most startling discoveries is that specifically among those lacking E4 was a genome-wide significant association, with the association peak being in a region of the promoter of the gene cancel-1, which is adjacent to the gene for MAPT. And you can see that in those with E4, the association was not really apparent at all. So a further analysis of this information showed that one of the genome-wide significant SNPs, particularly the one that's in the promoter of cancel-1, EQTL analysis showed that it impacts alternative splicing of one of the exons, exon 3, in MAPT. And importantly, the risk allele, if you will, the effect allele of the cancel-1 SNP was associated with reduced AD risk. And when that SNP is present, when that variant is present, it splices such that MAPT exons 2 and 3 are transcribed, and in the isoform in which it's transcribed, it's been shown to be associated with reduced risk of AD. So here is a list of genes that have been implicated with Alzheimer's disease through a variety of approaches. And all of these genes have in common that they are either tau itself, as in the case of MAPT, involved in tau processing, or in neuronal signaling. Many of these genes have been identified by GWAS, but others have been identified through other approaches. And I'm going to focus on 2 of these, plexin A4 and ACAT9, which were not identified through traditional GWAS. And these 2 findings happen to have been made by my group, so I have a lot of background information to provide. So a number of years ago, very early in the game of whole exome sequencing, we performed a whole exome sequencing study of AD in a very small number of African Americans. And what we identified were several SNPs that were associated on a very weak level, but then when we genotyped them in 2 large cohorts from the Mirage study and the Generation study, we found association with 2 missense mutations in the ACAT9 gene with AD risk. They promoted AD risk. We then attempted to replicate this finding in a much larger sample of African Americans from the Alzheimer's Disease Genetics Consortium, and indeed we replicated, and there have been subsequent studies that have confirmed. Now, what I want to point out is that these mutations are African American specific. They don't exist in other populations. Haplotype analysis showed that both of these variants are on the same background haplotype that occurs in African Americans at the frequency of 5%. But within that haplotype, those with one of the mutations or those with both of the mutations collectively account for 1%. So roughly 20% of individuals with this 5% haplotype have one or both of these mutations. Looking at these mutations in terms of where they are in the protein and whether or not they are conserved, both of them are located in the longer isoform, the ACAP 450 isoform. In the case of this particular variant, you can see that it's highly conserved through vertebrates, whereas this variant is certainly conserved within primates and higher mammals, but not in lower organisms. The point being is that it's very likely that these are conserved and important, and the mutations themselves would be deleterious. There have been a number of links established many years ago between ACAP9 and AD, but these findings didn't get as much attention. ACAP9 encodes an ACAP kinase anchor protein. It's been associated with the development of neurofibrillary pathology, and in one study, it was shown to be upregulated in the hippocampi of 80 patients. Now, what's also interesting is that this longer isoform ACAP 450 is a microtubule stability protein, which is what tau is as well. Subsequent to this finding, my colleagues and I performed a series of functional studies to see if we can link these mutations to the disease itself. And to carry this out, we conducted studies using lymphoblastoid cell lines that were selected from 80 cases and controls with and without the mutation. And we infected these cells either with the APP Swedish mutation or one of the mutations of tau. And we performed a series of protein measurements by ELISA of APP, A-beta 40, tau, and p-tau. And tau and p-tau were measured before and after treatment with rolopram, which is a phosphodiesterase type 4 inhibitor. And then we analyzed the ratio of A-beta 40 to APP, phosphorylated tau to tau, using a random effects model, comparing those with and without, the cells with and without the mutation, in models that adjusted for AD status, ApoE genotype, and a random effect. So in brief, what we found is that AD itself was not associated in these particular cell lines with the A-beta 40 APP ratio or the rolopram treated or untreated p-tau-tau ratio. And ApoE genotype was not associated with the difference in these ratios. And the mutation itself was not associated with A-beta 40, or the ratio of A-beta 40 to APP, or in the cells that were not treated with rolopram, looking at p-tau-tau ratio. However, ACAP9 was significantly associated, the mutations were significantly associated with the p-tau to tau ratio in the cells treated with rolopram, with higher phosphorylated tau in the lines that have the mutation. And you can see that a little more clearly here, that when you compare among those who don't have AD, you see a significantly higher ratio in those with the mutation. And if we compare the cells from persons with AD, we also see a higher ratio with those with the mutation. Next, we conducted a series of proteome experiments by mass spec looking at post-translational modifications of tau. And these were derived from the Rolopram treated lymphoblastoid cell lines. And you can see that there were at a number of residues significantly higher modification in those with the ACAP9 mutation. And in particular, including those that are known to be associated with AD, the ones here in red. I want to turn to now another gene that we identified in the Framingham heart study by a GWAS, but here what was unique is instead of using Alzheimer's disease status as the outcome, we created a quantitative measure based on liability of having liability for disease, what we'll call propensity. And on this scale, there is a threshold by if you have enough of that liability, you will manifest disease. And what you can see is those who barely cross the threshold would tend to have an older onset disease, and those with the youngest onset of disease, we could say, have the greatest liability. Conversely, looking at those who are unaffected, those with the highest liability are the youngest individuals. In other words, those with many more years in which to develop disease, whereas people who survive to very old age and don't develop AD are presumed to have low liability. And just to illustrate how we apply this, here are a few examples of a pedigree. All of the red shaded symbols are persons with AD, and based on the onset of the affected parent of this proband, who is at age 60, given the high density of cases and the onset of the affected mother, one would predict with a probability of 89% that this person will develop Alzheimer's. Once you start removing affected subjects from the pedigree, the probability goes down. And when you have no affected members in the pedigree and the person lived to be 80 years old, the parent, I should say, we would predict that that person has a low probability of developing AD. So we use these derived measures for the Framingham study, which is a family-based study, which is why we were able to do this. And here you can see that in the Framingham heart study cohort, we had 61 incident cases and over 2,800 controls. We also used another family-based study, the NIA load study, in order to confirm findings. And the primary finding or novel finding that we identified in the Framingham heart study cohort was association with genome-wide significant with several variants in the gene plexin A4. When we then tested those variants and other variants in this gene in the NIA load sample, we did see significant association, but with SNPs that were slightly further away. And when we combined them, we had essentially two peaks. And if you look here, you can see that these two variants, the one in the Framingham cohort and the one in the NIA load cohort, are located in an intron in the portion of the gene that encodes the SEMA domain. And interestingly, these two variants, where they are precisely located, flank an alternatively spliced region that creates a shorter isoform of plexin A4. So the full length is TS1 and this shorter one TS3. When we then looked at this mutation in other cohorts, we from the ADGC, we identified significant associations, not at the genome-wide level, but at other locations in the gene. And here I'm just highlighting where in European ancestry, in Japanese, in the region that encodes the cytodomain, and one other in the same intron that was significant in African Americans. Now, these variants across the gene are not in high linkage disequilibrium, and that suggests the possibility that there are multiple functional variants. We next set out to study the effects of the isoforms on tau phosphorylation. And we examined in neuronal cell line, SY5Y cells with a tau mutation, the effect of the amount of tau phosphorylation. And you can see here in those with AT8 that there was a significantly higher phosphorylation among those with TS1, but not TS3. And when we also looked at this effect in primary neurons from rat hippocampus, you can see here that there was significantly higher phosphorylation in those with the TS1 isoform, or specifically linked to the TS1 isoform. And the difference was significant for the TS1, the longer, versus the TS3 isoform. So here, the isoforms appear to show a differential effect on tau phosphorylation in cells, neuronal cells, and in rat hippocampus neurons. Next, we performed qPCR to measure the isoform expression in brains from 80 cases and controls. And you can see that there was a significant correlation of 0.75 with the clinical dementia rating measured in these individuals prior to death. And specifically looking at the association with AD in the TS1 isoform, especially in the late stage AD versus the control brains, there was a significant association, and less so for those with TS3. And want to point out that this is a relatively small sample, 5 controls in 14 cases. So looking at where plexin A4 sits in the pathway related to tau, here's plexin A4, here is one of its ligands, SEMA3A, and you can see that this region, or this pathway, leads to microtubule reorganization and ultimately degradation to form tau. And there are prior studies that have associated these two genes that are closely in the network to plexin A4 with AD related to tangle formation. So, and again, in summary, TS1 and TS3 isoforms of this gene may have different effects on tau phosphorylation. So in summary of the plexin A4 findings, we identified a novel gene in different SNPs in different family data sets localized in the sema and cyto domains, suggesting allelic heterogeneity, and predicted to have different functional consequences on the isoforms. We performed cell experiments in primary primary cultured neuronal cells, as well as in rat hippocampus, suggesting that there is an event upstream leading to tau phosphorylation, noting that there was no effect on APP processing or A beta production. And when studying the effects of these isoforms in brain, we showed that TS1 in particular, but both isoforms were correlated with AD-related pathological traits. And TS1 is highly correlated with clinical dementia. And TS1 is significantly overexpressed in AD brain. So in summary of this presentation and in preview to highlights of this session, MAPT region has been implicated in Alzheimer's disease, and as you will learn, in Parkinson's disease, with a much stronger association in persons lacking ApoE4. Tau, we know, is central to other dementias, CTE, frontal temporal dementia, and ALS. Tau has multiple AD-related functional roles, including in RNA metabolism via RNA binding proteins in stress granules. And there are many AD-associated genes involved in neuronal signaling and tau processing that are upstream of tangle formation and hence may make these potential therapeutic targets. And finally, as you will hear in another one of the talks in the session, tau now, using PET imaging, may have a role or may be used in precision medicine, both for diagnostic classification and prognostic prediction. I want to thank all the investigators and participants of the ADGC and the Alzheimer's Disease Sequencing Project who contributed in some way to the work that I presented today. In particular, I want to acknowledge my colleagues at Boston University. Much of the functional experiments were performed by Tunea Kezu and Carmela Abraham, and then a whole host of other individuals I work with performed all the analysis experiments. I also want to recognize the other co-leaders of the ADGC and all of the grant funding that supported this work. And I want to thank you for your attention and hope you look forward to seeing the other sessions in this symposium. Thank you.